Welcome here in the Blauwe Zaal and welcome to this lunch lecture by Studium Generale on emerging viruses. It's great to see such a big turnout today. Even after 3 years almost of living through a pandemic, it means, I guess, that you are still not tired of hearing about viruses. And I think you made the right choice because, well, fortunately, it seems that COVID-19 is now largely behind us. But unfortunately, that doesn't mean that there is no danger for a next pandemic simply just because we've just had one. And even though COVID-19 might have originated in China, the next pandemic could very well be on our own doorstep. Because, this is all very positive news. No, but it is a fact that the Netherlands are one of the largest hotspots for animal disease outbreaks in the world. Perhaps some of you remember outbreaks of bird flu in 2003, Q fever in 2007. These not only affected a large amount of animals, but also thousands of people fell ill. And also we've had swine fever, BSE, mad cow disease, and foot and mouth disease. Influenza viruses, they always manage to stay one step ahead of human and animal defenses. And evolution is very fast in the pressure cooker that we have in the Netherlands here. What could we do to reduce the impact of new pandemics or hopefully prevent them from happening altogether? Well, we have a true expert with us today, Ron Fouchier. He is professor of molecular virology at the Erasmus Medical Center in Rotterdam, also deputy head of the Department of Viroscience. Started his career studying HIV, then moved on to influenza viruses and is now a leading authority in bird flu viruses. And his two research groups study the ecology and evolution of influenza viruses as well as their public health impact. He is here today to discuss with us how viruses will take over the world, but also how their spread could be contained. So please welcome Ron Fouchier.
Thanks, Marla. It's a pleasure to be here, and I'm also happy to see that you're not yet bored of viruses. This is the picture that Marla referred to about Netherlands being one of the hotspots for emerging infections. In yellow on this map are the hotspots globally. And when we're talking about emerging infections, we're really talking about zoonotic infections. The vast majority of new infections that we see in humans originates from animals. And when animal viruses jump into humans, we call this a zoonosis. And so for these zoonoses, we need two things, is humans and animals. And the yellow areas are really the areas where we have most humans and most animals condensed in one spot. And also the right environmental conditions. So I borrowed this picture from Marle about the Netherlands. This is a picture of the Netherlands. In the Netherlands, we have about 100 million chickens at any point of the year, 11 million pigs, 4 million cows, 850,000 goats, and 500,000 goats. What Marle did not draw in this image is that we also have 17.5 million people, and with that, we are one of the most densely populated countries in the world, who also tend to like animals. About a quarter of our households have cats, and about 1 in 5 families have a dog. And so plenty of animals and plenty of humans. And also the land use is accompanying all this. Of course, we have enormous amount of agriculture areas to house all these animals. We have large areas of cities to live in. But we also have a big chunk of natural reserve areas, for instance, woods and wetlands associated with large bodies of water that we have. And we are very fortunate that in those nature reserve areas, we have enormous numbers of wild animals as well. Any point in time, we have 20 million birds breeding in the Netherlands. And in the winter season, we also have millions of birds migrating through our country. And so this is why the Netherlands is one of the hotspots in emerging infectious diseases. And so when I moved to Rotterdam, it was in 1998, shortly after an outbreak of bird flu in Hong Kong. And since that time, there really hasn't been a dull moment in the Department of Viroscience with new outbreaks that we need to deal with almost every year. Sometimes these outbreaks start in the Netherlands, like Marla pointed out, 2003 bird flu was one of the biggest bird flu outbreaks ever recorded globally. And 2007, Q fever, again, the biggest Q fever outbreak ever recorded globally at that time. But also we see sometimes viruses that emerge somewhere else and then cause a pandemic. 2019 COVID, exemplary. But of course also the flu pandemic of 2009, which you probably remember. Sometimes these outbreaks are transmitted by mosquitoes, such as Usutu virus in wild birds. Sometimes these infections are occurring elsewhere in Africa, Ebola virus outbreak, and then sometimes we see patients in our hospitals in the Netherlands, individual cases of Ebola entering the Netherlands. What you maybe do not realize is that even after COVID, we already had two major events that you may not be aware of. One is the arrival of West Nile virus in the Netherlands, which is a mosquito-borne disease spread by birds and causing encephalitis in humans. We've seen few cases in 2020, virus again detected last year. And of course, outbreaks of monkeypox globally that have been in the news a little bit more. But today I will focus on one group of viruses that is depicted here in 2013, 2009, 2003, and 1997. These are the influenza viruses, or flu. In fact, these outbreaks are listed here as single years, but most of these outbreaks have actually continued on until the present day. About influenza viruses. So the natural host of influenza viruses are wild aquatic birds, including ducks, geese, and gulls. And this time of the year, in the winter, early winter, late fall, if you catch a duck here in the Netherlands, there's a big chance that it will have an influenza virus. The virus is shed in large amounts in the feces of these birds, and so the viruses are transmitted through a fecal-oral route. Humans do not really contract viruses from wild birds directly because we don't come in contact much with wild birds. But poultry certainly does, or pigs, and certainly if that poultry or pigs are roaming outdoors. The Netherlands likes to have outdoor ranging chickens and pigs, but that of course increases the chance that viruses will jump from wild birds into our domestic animals. If birds defecate in surface waters, marine mammals may contract the disease as well. Humans generally get flu from poultry or pigs, not from wild birds directly. This is a diagram of an influenza virus particle. This particle is about 100 nanometers in size. It contains a lipid bilayer envelope, which is derived from the host cell. Inside the particle is RNA, 8 pieces of RNA, coated with nucleoprotein, and each piece of RNA is connected to a polymerase complex consisting of acidic polymerase, basic polymerase 1, basic polymerase 2 to replicate the RNA when the virus enters the cell. On the outside of the particle are two spike proteins, hemagglutinin in blue and a neuraminidase in red. The hemagglutinin protein is used by the virus to attach to receptors on the host cell. So it attaches to epithelium of our airways or to the epithelium of the intestinal tract of birds. The hemagglutinin binds to sialic acids, which is the terminal residues of sugars of glycosylated proteins on that epithelial cell layer. The neuraminidase protein cleaves those same receptors off in the process to be released from infected cells. So to facilitate release, the neuraminidase cleaves the receptor. In wild birds around the world, we see 16 variants of hemagglutinin and 9 variants of neuraminidase. And these can be found in all combinations, so in principle, 144 variants in wild birds of the world. And this is how we give these viruses names. So I will be talking today about H5N1, H7N9 primarily. These subtypes are biologically relevant because they are determined with antibodies. If a host, if you have been infected with an H1N1 influenza virus, you mount an antibody response to H1 and to N1, and that might protect you from subsequent challenges with H1N1 viruses. But it will not protect you against challenges with H2345 or N2345. And so these subtypes are biologically relevant. There's one more remark to be made about these bird flu viruses. All these 16 hemagglutinin subtype viruses are low pathogenic in wild birds. So these wild birds have co-evolved with their virus for thousands of years, and wild birds don't suffer at all from these virus infections. Also, when these viruses are now transmitted into poultry, in general, poultry does not suffer much. They may experience a little bit decrease in egg production or weight gain, but in principle, these viruses are non-virulent in chickens. However, two subtypes, H5 and H7, when these are transferred into poultry, these viruses can evolve, they can mutate to become highly pathogenic avian influenza viruses. And these viruses replicate systemically in chickens, cause systemic disease that kills the chicken in about 24 hours. But this only happens to H5, H7 viruses, and it only happens in poultry. Over the years, we've seen a dramatic increase in the number of events of evolution of viruses to these highly pathogenic forms. Here in orange line, you see the increase over the last 50 years of the occurrence of highly pathogenic viruses around the globe. And this is mirrored by the increase in poultry production systems around the globe. So the fact that we're producing more poultry means we're also producing more of these dangerous forms of bird flu viruses. So one more slide in general about emerging viruses before I move on specifically to flu, just to make the point that what I will be telling you today about influenza really applies to a very large range of viruses. My lab has been working on influenza viruses, so the flu viruses, but also on paramyxoviruses and pneumoviruses and coronaviruses. And for all of these virus families, the same story can be told, that there are hosts in nature that we call reservoir species, where there is a massive diversity of viruses circulating that have co-evolved with these hosts for thousands, if not hundreds of thousands of years. These virus families also occasionally jump over into other species, including wild and domestic species. And when these viruses enter domestic species, they may also transmit into humans. And what we see in humans is that several of these members of these virus families have caused pandemics in the past. You're all now experts on SARS-CoV-2, the virus that caused COVID in 2019. What you may not be so familiar with is that there are 4 other coronaviruses that have caused pandemics in the past as well. And each of these coronaviruses can be traced back to bats, and sometimes we also find intermediates in domestic animals. And so we can infer from this that all of these viruses that currently cause winter outbreaks in humans, currently we have outbreaks with these seasonal viruses as well, right now, this week. And these can be traced back to animal viruses in the past. In particular, OC43, for instance, we can calculate by looking at the genetic material of the viruses that this pandemic may have occurred a little over 130 years ago. And similar viruses as OC43 can still be found in cows today. We currently still have outbreaks of zoonotic viruses, so viruses transmitted from animals, in particular MERS-CoV, which is circulating in camels in the Middle East. It's causing zoonotic infections, and of course, we are trying to prevent this virus from triggering next pandemic. Same story can be told about the paramyxo and the pneumoviruses. There are 8 of them, endemic in humans, causing outbreaks every winter season. All of these can be traced back to domestic animals and to wild animals. Latest pandemic here, human metapneumovirus, virus that was discovered in my team about 20 years ago. This virus closely related to viruses of poultry and wild birds, and we estimate that this pandemic might have happened a little over 200 years ago. But today I will be talking about flu, and the reason to talk about flu is that both these zoonoses and these pandemics occur much more frequently than for the other virus families. For flu, we see pandemics on average every 30 years, and we see hundreds of zoonotic events by various influenza viruses every single year. And so you can imagine that if we have these zoonotic outbreaks in our country or anywhere else in the world, that we get questions from the health authorities whether we are facing a new pandemic. And every time we get this question, we don't know what to answer. And the problem is that we have no knowledge of what makes a virus transmissible in humans. Because the key difference between a zoonotic virus and a pandemic strain is its transmissibility between humans. If a virus is not transmissible, then the zoonotic event may be very ugly for an individual case, maybe one or two family members, but it's not a public health threat. It only becomes a public health threat if this virus acquires the ability to transmit between humans and spread around the globe. But this happens on average, as I said, every 25 to 30 years. The last one, 2009. Most of the students here were young and were certainly infected with this virus and perhaps even very severe infections. The fatality in this pandemic was relatively mild, so this was a mild pandemic, and this is why you probably have forgotten about it already. Milder than the pandemics of 1968 and 1957. But they are not always this mild. In 1918, we had the Spanish flu, killing in the pandemic years 50 million people in a world population that is 4 times smaller than it is today. And so these pandemics can be extremely severe, and in fact, the good thing was that Spanish flu ended World War I, because there were no more people to send to the front. The reason, by the way, that this pandemic was relatively mild is that this virus has the same subtype as this one. And this meant that everybody in 2009 who was born between 1918 and 1957 and had a primary infection with H1N1 already was actually protected to severe disease and death in 2009. And so the risk group, in general in pandemics, elderly people, were particularly protected in 2009. And the 200,000 deaths were primarily attributed to young children. But these viruses spread very, very fast. In 2009, viruses first detected in Mexico and California in April 2009, and within a couple of weeks, the virus spread to over 40 countries in the world and was essentially unstoppable. And the reason is that these flu viruses are transmissible through the air, through large and small droplets, like coughing, sneezing, talking, etc. This is a real picture, not an artificial picture, just taken with a sensitive camera and appropriate light source. And this is what typically happens if you cough, why your mom and dad will tell you to hold a hand in front of your mouth. Or these days, we have to cough in our elbows. Influenza viruses are well known to transmit both by these large droplets and by these smaller droplets. Some viruses are primarily spread by the smaller droplets, some viruses preferably by the larger droplets. With COVID, we think it's primarily, not exclusively, but primarily these larger droplets. But for flu, it's both the smaller ones and the bigger ones. The flu community, including my lab, has set up animal models to study transmission. And the system is very, very simple. We have ferrets that we inoculate with influenza virus. And the next day, we put a second ferret in a cage next door. And these two cages are separated by 2 steel grids, about 10 centimeters apart, so that these ferrets, they can look at each other and talk to each other and flirt with one another. But they cannot touch. And that means that if this ferret becomes infected, virus must have traveled through the air. Very old-fashioned system. We use ferrets because the airways of ferrets are very similar to the airways of humans anatomically. And also the distribution of virus receptors from the tip of the nose to the lower alveoli is It's very similar distribution in humans and ferrets. And as a consequence, ferrets are similarly permissive to influenza viruses as humans. And we've actually noted in pandemics of the past that when humans transmit influenza viruses through the air, so do ferrets. And so this is how the ferret model was designed. And so now we can start to address what is really needed for a virus to become airborne in a mammal, just like a ferret. And we started investigating this with H5N1 bird flu virus that was first identified by Alp Osterhaus in Hong Kong in 1997. It was found that this virus killed humans. 18 people got infected in Hong Kong, of whom 6 died. And Alp linked this virus to bird flu viruses that were spreading in the live animal markets of Hong Kong. Many people did not believe Alp when he told this story, because at this point in time it wasn't thought that bird flu viruses could infect humans at all, but Alp showed differently. The Hong Kong authority decided to kill everything with wings in the city of Hong Kong, and that stopped the outbreaks, of course, in poultry, because there was no more poultry, but it also stopped the outbreaks in humans. And we thought that we had averted a pandemic at that point in time. What was not known was that this virus actually was originating from mainland China, where it was not reported, and continued to spread even after 1997. And from 2004 onwards, out of China, the virus was transported into other countries in the area through poultry trade, and later also through wild bird migration. Also in China, a lot of birds are kept outdoors, which means they come in contact with migratory birds. Migratory birds can start to migrate and carry the virus along. And in many of the countries where the virus now landed, culling of the poultry was not an option, because poultry is a primary source of protein in some of these countries. And as a consequence, this virus is still zoonotic today in many countries in the world, and has caused well over 1 billion poultry deaths. It's also been transmitted to at least 20 species of mammals, including to humans. It's important to note that this virus is the same virus, essentially, as the virus as we are having today in the Netherlands. The outbreaks that we have over the last years are direct offspring of the viruses in China in 1997. My lab also studies ecology of avian influenza in wild birds, and the last few years have been devastating years for birds and for the ornithologists. This year, we had massive outbreaks on colony-breeding seabirds in the Netherlands, including these tern colonies that were completely wiped out due to H5N1 bird flu. Last year, we had massive die-offs of geese. Associated with these introductions with wild birds are, of course, outbreaks in poultry. This is a listing of Veterinary Institute in Lelystad, recording well over 1.3 million chickens culled just in the last 2 months alone in the Netherlands. So, devastating outbreaks of bird flu. And so you can imagine that with outbreaks like this, so this virus is now also spread to North America, and this year for the first time ever into South America. So this virus is now globally present. And the key question is, of course, can a virus like this become airborne and cause our next pandemic? To understand what must happen, we should review the literature of what we know about avian influenza and human influenza viruses. Avian influenza viruses replicate in the intestinal tract of birds, as I mentioned, and they bind to alpha-2,3 linked sialic acid. So these are the terminal residues of glycosylated proteins on the epithelium of the intestinal tract of a bird. And the viruses have co-evolved with this receptor for thousands of years. Human influenza viruses, all pandemic and epidemic strains, prefer to bind to alpha-2,6 linked sialic acids. Very small change, but significant. And this receptor is abundantly expressed in the upper airways of humans. Humans also have the avian receptor, but primarily in our alveoli. And this might explain why these bird flu viruses kill humans. If by accident you get the virus in your alveoli, you will get a pneumonia. But this might already also explain why this virus is not transmitted, because it's not so abundant in the upper airways, perhaps. Second key difference is the optimal temperature for replication. The body temperature of a bird is about 42 centigrade, and the polymerase of influenza viruses have perfectly tuned to operate at this high temperature. But when viruses now transmit into humans, in particular in the upper airways, temperature is much lower. It's about 33 centigrade. And what we see is that all human viruses prefer this very low temperature. And so apparently, when viruses cause a zoonosis and then become pandemic, this change facilitates virus replication. We also know from the pandemics of the last century, we had 4, which genetic changes in the viruses drive these phenotypes. And there's really only a few genetic changes that we observed in pandemics of the last century. And so we can now take these genetic changes and insert them in an H5N1 bird flu virus to see if that would make the virus transmissible. And that's what we did. So we took a H5N1 bird flu virus from Indonesia, engineered it to have receptor binding mutations, polymerase mutation that we know from all pandemic viruses, but this virus is not yet airborne in the ferret model. So we're still missing knowledge on what makes a virus transmissible. But we can now take this virus and put it in the nose of a ferret, let it replicate, and then physically move it into the nose of the next ferret. And we repeat this a couple of times. And what we're doing here really is an experiment of natural selection according to Darwin's evolution theory. These viruses mutate, and they naturally mutate. Viruses mutate a lot. And we get natural selection of viruses that are better adapted to the upper airways of a ferret. And what we're creating is a mix of additional mutations that apparently allow these viruses to outcompete the original one. And we can now take this mix of viruses and test whether there might be some that are now airborne. And the answer is yes, that happens. So when we inoculate a genetic mix of mutants into the ferret, put ferrets in the cage next door a day later, we see that this virus is transmitted to 3 of the 4 pairs. When we take the virus from one of these pairs and put them into the next series of ferrets, again, this virus is airborne. And so this experiment is important for 2 reasons. One, it shows that the H5N1 virus can become airborne in a mammal, not necessarily in humans, but certainly in mammals. Very important information, because some people, after 25 years of not seeing a pandemic, did not believe that this was possible. But in principle, it's possible. But more importantly, we can now isolate the viruses back from these animals and see what really happened to them and understand what makes this virus airborne. And to run a very long story short, the number of changes is very, very limited. There's really only 3 traits, 5 mutations that can drive the airborne transmissibility of this virus. We inserted 1 mutation that affected temperature optimum, and it seems that this virus needed 1 more change to accommodate that lower temperature optimum. When we insert 2 mutations, we boost the replication of the virus in human cells at low temperature by more than 100-fold. We also engineered 2 mutations that we knew from all the pandemics in the HA. It appeared that we only needed 1 of those 2 that we engineered. But we missed another one, so the viruses acquired an additional change at the tip of the hemagglutinin, which is a sugar molecule. If you lose that, the virus attaches much better to the receptors. The phenotype that we missed was this stability phenotype. If you introduce these mutations, you destabilize the hemagglutinin in such a way that the virus is hardly infectious anymore, and you need an additional mutation to stabilize this hemagglutinin with respect to low pH. The entry of viruses in cells is pH dependent, and this low pH is needed to keep this virus infectious. And so really only 3 traits, 5 mutations that can drive airborne transmission of this virus. Now, this is important knowledge because now, if there are outbreaks ongoing in poultry, mammals, or humans, we can screen for these mutations, and more importantly, we can screen for these phenotypes. If we see these phenotypes, we should move aggressively, cull the animals, quarantine the people, make sure that this virus doesn't spread. Also, this is important because these H5N1 infections have continued to occur throughout the years. And so each of these human infections could be a natural selection experiment, just like our ferrets. These mutations can be acquired in real-life situations. And so each of those cases should be followed up and quarantined if needed. While we were doing this work, a new virus popped up, this time H7N9, again in China, causing many more infections and deaths than the previous one. But what is really surprising about this outbreak is that China was very forthcoming with information in this instance. They released virus genome sequence data the same day and offered the virus to other laboratories in the world in the same week, and they have never done that before. The reason was that they found that these viruses, these early human viruses, had the same mutations as in our airborne H5 virus, and they were afraid that we might be facing a new pandemic threat. And so they shared this virus with the world to investigate, including my laboratory, Mathilde Richard, who's an assistant professor in my team, who did this investigation. And we found that this virus indeed has mutation that allow the virus to replicate at low temperature. It's done by the Chinese teams, and also that this virus already binds to human-type receptors, also shown by the Chinese teams. The other three teams showed that this virus indeed is transmissible in a ferret model. The good news was that all of these three teams found that the virus was inefficiently transmitted in the ferret model. It took 4 or 5 days of exposure before the virus jumped, whereas with general pandemic strains, this happens within a single day. And also the virus only jumped in about 1 in 2, 1 in 3 animals. With a true pandemic strain, this happens in 100% of animals. So we're not quite there yet, and we think that this is due that this virus still doesn't have the stability mutation. We investigated. The stability mutation is easy to acquire, but it wasn't acquired in humans. Good news is that the Chinese authorities, when they saw these outbreaks continue, decided to vaccinate all their chicken. And so after 2017, we've not seen many human infections anymore because China vaccinated every single chicken in the country in 2017 to stop these outbreaks. And it's very well possible, therefore, that the Chinese, with doing so, averted a pandemic. We will never know, because we do not have a parallel universe where we can do the same experiment, but then without vaccination. But better safe than sorry is what the Chinese thought, and this experiment worked. Yeah. Last case is a case of, in the Netherlands, of H10N7 virus. It's really a very similar case as H5N1 in our laboratory, but now it happened on our beach. There's an outbreak in seals, spreading in seals in the Netherlands. And these viruses acquire the same mutations, and that then results in accumulated wisdom of what makes an influenza virus transmissible. And so now, from now on, in outbreaks, we can start to investigate which of these traits and mutations are found in viruses in the world. And so to conclude, I made the point that respiratory viruses will continue to emerge in humans and other mammals, and that there is currently little knowledge of what makes these viruses transmissible, which is really key knowledge to prevent, well, for our basic understanding, but also to assess risk and to better prepare or prevent pandemics in the future. For influenza viruses, we now know what the determinants are, and during surveillance studies, we can search for these traits. If we find these traits, we can cull animals or quarantine humans to prevent spread. But importantly, and also for this university, I think we need to do more research on more generic and broad pharmaceutical and non-pharmaceutical interventions, including, as we've seen in COVID, how important face mask, distancing, ventilation are. And some of these options are, of course, also of interest to this university. On the long run, some more radical action may be needed, such as changes in our farming practice to reduce the overall risk. Because currently we are really waiting for ugly outbreaks to happen in humans. We know that viruses are circulating in wild and domestic animals, but we do not respond until we see spread of virus in humans. And we need to change this behavior. We need to investigate viruses in wild animals, prevent the spread in domestic animals and stop them right there if this happens, and have active surveillance in humans to very early interfere before the virus becomes widespread and causes a pandemic. This way we might be able to prevent some pandemics, and if we cannot prevent the pandemic altogether, we need to have broadly active pharmaceuticals and non-pharmaceutical interventions available. With that, I will stop. Just to comfort you with the thought that we do this research in highly safe and secure laboratories only. That, of course, we cannot do this work in an ordinary laboratory, but we have very nice facilities for this work. And with that, I will stop. Answer questions.